Okay, let's talk about blood. We're going to do this a little bit different this time, and we're going to use this kind of whiteboard format as we go through. You can tell me how you like this compared to PowerPoint or something like that. But let's start talking about blood. And if you guys remember from anatomy, blood is a type of connective tissue. So as a type of connective tissue, that means it has to have both a matrix as well as some kind of a cell, right? So every connective tissue has a type of cell and a matrix. So bone has osteocytes plus that mineral calcium-based matrix. Well, for blood, the matrix is called plasma, which I think we've already mentioned here in previous lectures. And then, of course, there are blood cells. And blood cells are basically separated into three different types of cells. So you have red blood cells, and that's what we'll talk about mostly today. Those are the ones that carry oxygen, right? And then you have white blood cells, and those are going to be largely involved in immune function, and we'll actually have a separate discussion for the white blood cells, so we won't mention them too much in this lecture. And then we have platelets, and platelets are used for clotting, and we will spend a little bit of time today talking about the clotting cascade, what happens in clotting, and how all that works. But if we come back to the plasma, which is the matrix of blood, the plasma is largely composed of water. It's about 92% water. There are some proteins, about 7% proteins, and I'll just say that most of those proteins are albumin, and albumin is made largely in the liver. And then there are also, we could say 1% would be ions and other organic materials that are just kind of floating around in the plasma. So the formation of blood occurs largely in the bone marrow, and you'll probably remember that from anatomy. And we call the formation of blood hematopoiesis. So hematopoiesis occurs largely in the bone marrow of our longer bones in particular. And 25% of that is devoted to red blood cell production. And that's largely because, so that means obviously 75% is going to be white blood cells. And that's largely because the red blood cells live longer. So they live an average of 120 days, we would say is the average lifespan. And so because they last longer, less of the bone marrow production is consumed with red blood cells. White blood cells, on the other hand, are broken down quite rapidly. They get used up very quickly. Really, we're talking about hours, maybe to a couple of weeks at most for most white blood cells. Most of them only last a few days. And so the body has to spend a lot more energy producing white blood cells just to kind of keep up with the destruction that's occurring weekly. And what controls hematopoiesis, depending on what we're talking about, red blood cells or white blood cells. So we've talked about in the past that for red blood cells, it's erythropoietin. In fact, we just mentioned that in the last lecture. And you can see this poietin and this poiesis. You can see how they're the same kind of root word, which basically means formation or growth. So erythropoietin stimulates red blood cell production, and I think that we've already talked about. White blood cell production is a bit more complicated. We're just going to focus on two of the molecules that help stimulate white blood cell production. and those are cytokines. Sorry, that's supposed to be an O there, cytokines, as well as what we call colony stimulating factors, CSFs, colony stimulating factors. So those two are responsible for white blood cell production as well as some other molecules that we're not going to focus on. And the last one we mentioned was platelets. So platelet production is stimulated by something called thrombopoietin. And once again, you see the poietin ending just like you did in erythropoietin and hematopoiesis. And thrombo is kind of the root word that means clot forming. So because platelets are involved in clot formation, we call that thrombopoiesis. If we looked at, and we did this in anatomy, if you remember, if we kind of looked at the way that blood cells are made, we would start with some kind of like a pluripotent stem cell. And from this stem cell, we're going to kind of get every other cell in the body, every other blood cell. And so we could kind of have this cell can differentiate into really a multitude of other cells. We will mention briefly 6 of them here, and hopefully this is mostly review from anatomy. So if you remember, we have some cells that will eventually become red blood cells, and we'll skip some of the intermediate ones. The one that you really need to remember, this one is the erythroblast. If you remember, blasts were kind of the progenitor cells, and erythroblasts form reticulocytes, if you remember this from anatomy. Reticulocytes. And then the reticulocytes eventually form adult red blood cells. So reticulocytes are kind of immature red blood cells. And then we've got all of these lines, and they're all going to produce a multitude of white blood cells, but in particular myelogenous. Again, that's kind of review from anatomy, and we'll get into that a little bit more in the immune system. Then, of course, we've got to have a line that makes our platelets, and that basically comes from a very large cell that we call a megakaryocyte. And little bits break off of that, and that's how we get our platelets. So platelets are really just little bits and pieces of megakaryocytes that eventually break off. So this last line here would be what turns into lymphocytes, if we put down here. And again, we'll talk more about that in the immune system chapter. But lymphocytes would be the other kind of cell line here. So remember, this is kind of a review from anatomy. You get a pluripotent stem cell that can really become any type of blood cell. And then those cells differentiate either into a line that will become red blood cells, a line that will become myelogenous white blood cells like neutrophils, eosinophils, and another line that will become lymphocytic white blood cells. We'll talk about the differences between those in the immune chapter. And then one line that becomes megakaryocytes, and from the megakaryocyte we get platelets. So in this lecture, we're going to largely talk about red blood cells, and then spend a little bit of time talking about platelets and how they help form clots, and white blood cells will be for another lecture. So red blood cells, you'll remember from anatomy, that they have some unique features to them, and really they're designed to carry oxygen. I mean, that's That's kind of their whole purpose. They're not like a lot of other cells. Remember, they have no nucleus. They have no mitochondria. So they basically don't use up the oxygen that they carry in order to kind of keep their own cellular processes going. They're basically an empty sac full of hemoglobin. So hemoglobin is really the key element to red blood cells. And if you remember, they're also interestingly shaped. They have that we call biconcave shape, or what we've been talking about earlier is kind of a donut shaped, where the center of the cell is kind of thinner. And you guys saw this a little bit under the microscope when we did the tonicity lab. You could see that the center of that red blood cell looked different than the other than the outside part of the red blood cell, and that's because it's thinner in the center. And that's really important, because that's going to aid in diffusion. If you guys remember the principles of diffusion and Fick's law, the distance of diffusion affects the rate. So if you can take an already thin cell and make it even thinner in the center, that's going to really aid diffusion, because the oxygen that's inside the red blood cell is going to take less time to diffuse out, because the actual distance it has to move to get outside the cell is much lower in the center of the cell. So it helps with the diffusion. But remember, it also helps with bending, or we might say flexibility. It makes those red blood cells much more flexible, so that as they're moving through those tiny capillaries often are just the width of one red blood cell. And so it's almost like you have a bunch of red blood cells just kind of marching through that capillary, and they're just the right size. So having that kind of donut shape helps them to bend a little bit without breaking. And so it's a useful shape as well for bending, for flexibility. In fact, there are some pathological processes or diseases that actually change the shape of the red blood cell. And when that happens, it makes those red blood cells much more likely to burst or break open. And so you remember that 120-day lifespan that we talked about becomes much shorter in disease states that change the shape of the red blood cell. So the most common one is called sickle cell disease, and it changes the shape of the red blood cell from that kind of donut shaped into what looks more like a quarter moon shape, and that makes those red blood cells get stuck inside capillaries and causes not really clots, but it causes the backflow of blood, so that they kind of plug up small capillaries, and then you don't get blood flow beyond them, and it causes ischemia, or a lack of oxygen to tissues, which causes pain. But also this funny shape and the way that it's made makes it more easily destroyed. It makes it break apart or hemolyze much more easily. And so people with sickle cell disease end up having chronic pain, because these sickle-shaped cells are always getting stuck in capillaries and obstructing blood flow. And they also have anemia, which means a low red blood cell count, because they're always bursting their red blood cells because of the funny shape, and they're not able to produce them as quickly as they destroy them. And so often they require transfusions from time to time. So the shape of the red blood cell is really important, both for diffusion and for flexibility. And so it's really perfectly shaped to match its function. That's another one of those examples of how form determines function, or function is aided by form. Okay, so we were talking about red blood cells, and the shape and the function of red blood cells is important, but what's really important about red blood cells is what's inside the red blood cells, and that is the hemoglobin. And you'll see that I will often write hemoglobin in this way. HGB as a shorthand, so we'll do that from now on. But hemoglobin is the molecule inside red blood cells that is actually binding, or sometimes we say carrying, oxygen. So hemoglobin is composed of 4 subunits, or 4 protein subunits, and I think we talked about this in anatomy, so hopefully this is a little bit of a review for you, but there are 4 protein subunits for each hemoglobin molecule. So there are, in normal adult hemoglobin molecules, there are 2 alpha subunits and 2 beta subunits, and that's what we would consider to be normal adult hemoglobin. Now this is different from what you find in a fetus, for example. The hemoglobin molecule in a fetus is composed of 2 alpha subunits and 2 gamma subunits, and that's because that particular type of hemoglobin has a stronger affinity for oxygen, and fetuses live in a low oxygen environment, and so the hemoglobin they produce is really adapted to extract as much oxygen as possible from the blood that they get from the placenta. The blood in the placenta is already partially deoxygenated by the time it reaches the placenta, and so they need a different type of hemoglobin that binds to oxygen much more strongly. In adults, on the other hand, we live in quite a high oxygen environment, and so though we still need some affinity for oxygen, we still need to attract oxygen to our hemoglobin, so it still needs to attract and hold the oxygen. It doesn't need to attract it as strongly or hold it as tightly, because we live in an environment where typically we have more oxygen than we need. So remember, hemoglobin not only has to pick up oxygen, let's say at the lungs, but it has to then carry that oxygen. So let's say we have the lungs here, and we've got our blood vessel, and we've got our red blood cells, and of course we've got oxygen coming out of the lungs into the red blood cell, and that's important. It needs to pick up the oxygen there. But remember that when that blood vessel reaches, let's say muscle, it needs to drop off the oxygen, right? It's got to deliver the oxygen to the tissue. So hemoglobin not only needs to be able to grab oxygen and hold it and keep it, it needs to be able to give it up, to kind of willingly let it go when it reaches the tissues that need it. And when we get to the respiratory system, we'll talk about why it is that at the lungs, hemoglobin functions to bind oxygen, whereas in the tissues, hemoglobin functions to release oxygen. So we'll talk more about the mechanism, the physiology behind that when we get to the respiratory system. But right now, just suffice it to say, hemoglobin has to be uniquely structured to both bind oxygen at certain pressures of oxygen, and then it needs to be able to let it go. So in the adult body, we have kind of a weaker affinity to oxygen, meaning we let it go more easily in the adult. In the fetus, the hemoglobin binds much stronger to the oxygen, which means it's more stingy with releasing the oxygen, but that works well in the fetus that has very low activity levels and lives in a poor oxygen environment. So when you become an adult, your bone marrow has to switch the type of hemoglobin that it makes from this 2 alpha 2 gamma to a 2 alpha 2 beta. And just for comparison, sickle cell disease has a mutation, so it's still 2 alpha and 2 beta, but sickle cell has a mutation in the beta globulin, and because of that mutation, it causes the shape of the molecule to change, which then changes the shape of the red blood cell itself. So, for example, if we just kind of draw a little picture here, we could say that you've got an alpha here and an alpha here, let's say, and then we've got a beta here and a beta here, and of course they're all kind of bound to each other. And all of this together then would all be one hemoglobin molecule, all wrapped up together. And the real key to each of these subunits, so we have 4 subunits, right? 2 alphas and 2 betas. But the real key to grabbing oxygen is in what is kind of in the center of each of these subunits. So each of the subunits, each alpha and each beta subunit, they are all equipped with what we call a porphyrin ring. And if we pull that porphyrin ring out, we will see that in the center of that porphyrin ring is a molecule of iron. And so it's actually that iron that is attracting the oxygen. So the oxygen is attracted to the iron. So remember, oxygen is typically a negatively charged molecule, and iron is typically a positively charged molecule. So the electric force of that iron draws oxygen to itself. So what that means is that for each hemoglobin molecule, remember each hemoglobin molecule has 4 subunits. So that means each hemoglobin molecule has 4 porphyrin rings, which means that it combined 4 oxygen molecules. So each hemoglobin molecule can actually bind, pick up, and hold and carry 4 oxygen molecules. Now again, this is super efficient. So instead of each red blood cell only, or each hemoglobin molecule only being able to pick up, 1 oxygen and deliver it, we have a structure that allows for 4 oxygen molecules to be picked up and delivered. And of course, each red blood cell has, thousands and thousands and thousands of hemoglobin molecules. So each one red blood cell is able to pick up a ton of oxygen. So it's really quite an amazing design. Okay, since iron is such a important part of hemoglobin and really of oxygen transport, I wanted to kind of follow iron as it enters the intestine and becomes part of hemoglobin, all the way to how it is eventually broken down and removed from the body. Because this whole process of iron absorption, iron utilization, and iron removal or iron breakdown is involved in a lot of red blood cell pathologies. So a lot of the anemias that we encounter have something to do with either iron absorption or iron transfer or the use of iron within the red blood cell. So let's just kind of follow a molecule of iron. Let's say you eat a nice, tasty T-bone steak, and so that goes, into your intestines, right? And your intestines are going to absorb the iron from that steak. Let's say the iron is going to be absorbed by the intestine. So that's kind of step 1. You eat it, you consume it, whether it's from meat or whether it's from, certain vegetables are high in iron as well. Your body has to then be able to absorb the iron from whatever food that you have eaten. And by the way, iron is typically absorbed better if it is absorbed in the presence of vitamin C. That's just a little kind of health tip for you. So once it is absorbed into the blood supply, it then has to be carried to the bone marrow so that it can be used in red blood cell production, right? So if we kind of have let's say we've got our bone marrow here, and we've got to get that iron from the from the blood after it's absorbed to the bone marrow. We need some sort of molecule to actually transfer the iron. Iron does not transfer kind of naked in the blood. It actually binds to another protein that we call transferrin. So if you remember, right, Fe is the chemical symbol for iron. So a lot of the words for iron start with Fe, ferrin, ferrous, ferritin. So transferrin, trans is kind of transport, that's how you can remember that name. Transferrin transports the iron from your blood to the bone marrow, where it will eventually be made into red blood cells, right? So from the bone marrow, we're going to get our RBCs. So now the iron is inside the hemoglobin, inside the red blood cell. And that's going to last, we mentioned, about 120 days, right? On average, that red blood cell will live about 120 days. Well, what happens at the end of 120 days, on average, they start to get, worn out, maybe damaged, and eventually they go through the spleen. So the spleen acts as kind of a filter for the blood. So as the red blood cells go through the spleen is going to kind of get rid of old red blood cells. It's going to kind of destroy those old red blood cells, break them apart, and then kind of use what was inside the red blood cell. We don't want to lose. We don't want to lose the iron and the hemoglobin and all that. If we can reuse that, then we want to reuse that. So what eventually happens is, certain proteins and things that were used to make the red blood cell, they can kind of be reabsorbed into the circulation. But the hemoglobin that breaks down, so if we take that hemoglobin that was inside the red blood cell, right? So once the red blood cell is broken down, that hemoglobin turns into something called bilirubin. And bilirubin has a couple of uses in the body. So bilirubin can go to the liver, for example. So if we follow bilirubin to the liver, it will be made into bile. You can see how bile is part of that word bilirubin. And bile is used to digest fats. And we'll see that in the gastrointestinal lecture. Eventually the bile goes back into the intestines, which, interesting, is kind of where we started, right? And it will be excreted into your stool, your feces. And it is actually part of the reason why your stools are a dark color. Much of the dark color in your stool comes from the bilirubin that's being excreted there. And the bile that is not, or the bilirubin that is not not sent to the liver to become bile, eventually gets cleared or filtered through the kidneys. And so you will also excrete some bile in your urine, and a little bit of bile is normal. If there's too much bile, then that usually means that there's too much bilirubin being broken down, which means there's usually a problem here at the spleen, or there's a problem here at the red blood cell. So sometimes we check the bilirubin content of urine, and if it's high, then we worry about red blood cell destruction being too high. So you can see we lose bilirubin in the urine, and we also lose bilirubin in the stool. And that bilirubin came from hemoglobin, and that hemoglobin, of course, came from the red blood cell and was made from iron. So I've mentioned this term already, but I wanted to just kind of stop for a moment and talk about what happens when we do not have enough red blood cells in our blood. We call that condition, we call anemia. Now, anemia can result from several causes. I mean, on the one side, you can just have kind of a low, or maybe we could say poor production of hemoglobin, red blood cells, and on the other hand, you might have increased destruction of red blood cells, and that might be why you don't have enough red blood cells to go around. Or perhaps you're even just bleeding. You could be bleeding from a colon cancer, bleeding from polyps, bleeding from an ulcer. You could be bleeding in your urine, all kinds of places you could be bleeding from. And that's going to, of course, make your body deficient in red blood cells. So the definition of anemia is kind of low red blood cells. So if we want to give that a definition. And so in order for us to kind of determine what's the cause or what's the source of the anemia, what's going wrong in the body that's leading to a low red blood cell, we can look at the shape and the size of the red blood cells. So if we look at the size of the red blood cells, if they're too small, we call that microcytic. So micro, right, would be small. Cytic just means cell. So the cells are too small. And if we see microcytic red blood cells in a patient who has anemia, then we worry about some nutritional problems, like perhaps maybe they have a low iron, maybe they either don't consume enough iron, or maybe they're consuming enough iron, but their body is just getting rid of it too fast through bleeding. So blood loss, a low iron intake, those would all give us what we call a microcytic, a small red blood cell type of anemia. We could also have what we call a macrocytic. Macro means big, right? So we would have larger than expected red blood cells. And when we see that, it often points to a different kind of nutritional problem, either a folate or a B12 deficiency. So if you're lacking those two vitamins, you often will have a larger than normal red blood cell. We call that macrocytic. The last thing that we can look at as kind of a cause of anemia would be what we call hemolysis. So hemolysis is hemo, which means blood, and lysis, which means to break. So that's the breaking apart of blood cells. And that can be microcytic or macrocytic. But a lot of times what we'll do is we'll actually do a peripheral blood smear where we'll take a sample of your blood and we'll put it on a slide and we'll look under a microscope. And here we're looking really for the shape of red blood cells to see if they have that nice donut shape, or are they more sickle shaped, or are they more spherical. That would give us an idea that maybe there's a genetic problem with those alpha and beta subunits that are leading to a change in the shape of the red blood cell, which leads to an increase in destruction of the red blood cells, or what we call hemolysis. So often that's really helpful, looking at the shape of the red blood cells. Okay, well, let's shift gears from the red blood cell, which carries oxygen, to the platelets. And remember, the platelets are really involved with clot formation. So we're going to spend the next couple of slides really looking at how are clots formed, what are the steps involved, what are the other molecules involved. So I want to start with just talking about the kind of the big picture. So clots are formed when there is a damage to endothelial cells. So if you have damaged endothelium, right, that's the lining of the blood vessels, capillaries, veins, arteries. So if you damage the endothelium, the body's going to recognize that. The body is going to know that. We're going to see here in a minute there are certain chemicals that are released from the damaged endothelium, and that's going to signal the body that there's a problem here that needs to be fixed. So then the body's going to undergo really a 3-step process. So there's a 3-step process to clot formation, okay? And the first step really involves the platelets, which we've mentioned earlier. So the first step, I'm sorry, that's going to be the second step. We'll come here to platelets in the second step. The first step is vasoconstriction. So, I mean, imagine that you have broken, a small artery, let's say, or even a capillary, the first thing that your body wants to do is limit blood loss, right? So the way that it limits blood loss initially is by vasoconstricting. So if you can constrict that blood vessel, you will begin to shunt blood away from that broken blood vessel, and you can hopefully limit some of the blood loss. So that's an important first step. The second step, which I alluded to, would be platelets aggregating. And I have mentioned this before. In fact, we talked about this platelet aggregation, if I can spell here. Platelet aggregation means that the platelets are sticking to each other. And so those platelets kind of attach themselves to the broken area of the blood vessel, and they begin to clump together as they stick to one another. And that becomes kind of a temporary clot over that broken endothelium. And this is why I mentioned to you both last year and here recently that when people are having a heart attack, which is most of the time caused from a clot in one of the coronary arteries, that the first thing we do is we give them aspirin. And aspirin is an anti-platelet drug, which means it actually prevents the platelets from sticking to each other. So if you can prevent platelet aggregation, then you can prevent clot formation, because platelet aggregation is what happens first after the vasoconstriction. Okay, once you get vasoconstriction to try and limit blood loss, and then you get platelets sticking together, what we call platelet aggregation, that's kind of a temporary plug. Sometimes we call that a platelet plug. We're then going to end with a true clot. So we're going to get true clot formation. But that's actually the last step. And that's because it takes some time. You've got to call several factors to the area. And it takes some time for a true, more permanent clot to form. Okay, so let's go through this process. Let's say we start with damaged endothelium here. That's kind of what started it all, right? So that damaged endothelium leads to some collagen that is exposed. And it's really that exposed collagen that becomes a trigger then for several things to happen. So one thing that happens is it triggers the vasoconstriction, which we've talked about. The other thing that it does is it actually releases special platelet factors that cause the platelet plug to form, that make the platelets actually more sticky. So these platelet factors, which are released from the exposed collagen, actually cause platelets that are floating by that exposed area. There's certain chemicals that are released there, and they cause the platelets to become activated, in which the platelets become more sticky. They begin to express certain molecules on their surface that allow them to bind more easily to other platelets. So that exposed collagen causes the vasoconstriction, plus the platelet aggregation, which forms that initial platelet plug. At the same time, however, a couple of other things are happening. So because that endothelium has been damaged, there's some what we call tissue factors that are also released, and those tissue factors are going to start what we call the clotting cascade. Okay, they're going to signal the release of several other proteins that are going to eventually form the final clot, the step 3, right? So this is step 1 and this is step 2, and this clotting cascade is going to eventually lead to step 3. I'll just say that the exposed collagen also kind of triggers certain molecules within the clotting cascade, and so you got kind of a double stimulation there for the clotting factors. The clotting factors, the clotting cascade is going to eventually lead to formation of what we call thrombin, and we'll see this here in a minute. Thrombin is one of those enzymes, if you remember when we were talking about the renin-angiotensin system, and we said renin helps convert angiotensinogen to angiotensin, so it was kind of a converting enzyme. Well, thrombin also functions that way. Thrombin is going to act as a enzyme of sorts to convert fibrinogen. So there's that ogen that we talked about, which was kind of the suffix on words that signify it's an inactive form. So fibrinogen was always available in the blood, but it was not active until thrombin interacts with fibrinogen, and fibrinogen becomes fibrin. Now, fibrin is super important because fibrin is going to be the molecule that actually leads to the true clot formation. So once fibrin is available, that platelet plug becomes a true clot. That's step 3, right? And then eventually that clot will dissolve and the tissue will repair. But that takes some time. So you can see how that exposed collagen, that injured endothelium, leads to all of these 3 steps that we have here. If we look at the clot formation, especially the platelet plug formation, we would say that that's a positive feedback system, because what's happening if we have, let's say we have kind of a damaged endothelium there, and we get that damaged endothelium secretes, we said platelet factors, and the platelet factors call more platelets to the area, right? So you're going to begin to plug up that injured area. But what happens is when those first platelets arrive, they also produce platelet factors. And those platelet factors will then call more platelets to the area. But of course, when those platelets reach the area, they will also become activated and they will release their own platelet factors, which will of course call more platelets to the area. And you can see how this becomes really a positive feedback loop. This platelet formation. So it's another rare example of positive feedback in the body. And eventually what you end up with is this platelet plug. And that's good for a temporary, repair of that injured endothelium, and it will help keep blood from leaking out. But it's not a permanent solution. It's really only temporary. So we still need step number 3, which is clot formation. So we'll see that here on the next slide. So clot formation is going to be really a complicated process, and we're not going to go into all of the factors involved in platelet formation, in clot formation. We're not going to go through all the molecules involved. There are several of them. There are several what we call clotting factors. So I'm just going to say clotting factors are made and are activated, okay? So we'll say they are made and they are activated. So most of the clotting factors are made in the liver, and this is why people who have liver problems like cirrhosis, for example, they have trouble forming clots because they are not able to produce clotting factors at the typical rate. So all of these activated clotting factors we mentioned lead to thrombin. So you do have to learn about thrombin. So all of the clotting factors lead to thrombin. And thrombin, of course, turns fibrinogen into fibrin. So we mentioned that, fibrinogen. So you need thrombin to do that. And then fibrin, we're going to see, acts kind of like a mesh. So fibrin will kind of come, and it kind of forms a mesh. Let's say we had our damaged endothelium here, right? And we had our platelet plug in place. So we had a bunch of platelets there, kind of plugging it up. Fibrin then comes, once it's been activated by thrombin, and thrombin is kind of activated from all of these other clotting factors. So once fibrin is activated and becomes aware of the problem, it begins to form this almost like a net, really like a meshwork. And what happens next after the platelet plugs, the fibrin comes and kind of covers over the platelets and forms a mesh, is you get red blood cells that are kind of floating by, right? They've been floating by. But now that there's this kind of sticky net or meshwork, these red blood cells then kind of get stuck inside this meshwork. And now you get a true clot forming that has both fibrin and red blood cells that kind of get caught in the fibrin, and that's able to completely cover over and seal over that damaged endothelium. And this is really the more permanent clot that can stay in place until the body finally is able to repair that area. Okay, and I think that's going to be really enough for this discussion of blood. I have uploaded for you a link to a short Khan Academy video that talks about blood typing that should help you with your case study. Make sure you take a look at that before you do the case study. I think it'll be really helpful, especially if you're not familiar with blood typing and how that works. Of course, if you have any questions, you can always send me an email and I'll be happy to respond as well.